So I help myself with the video Excitation Contraction Coupling by student 4AP. Please like the original and subscribe to original. Okay, this video is, I like this animation, but as you can see it's 1 minute 42 seconds, so we'll stop quite frequently because there is a lot of information here. Some of those information we already got through, but I suppose it's quite hard, so I will try to explain it one more time.
Know that excitation is how the nervous system stimulates an action potential in the muscle fiber sarcolemma.
Alright, I will slow it down.
For a skeletal muscle to function, it must contract, which means it gets shorter.
It's too much.
When a muscle gets shorter, the skeleton moves at the joint it crosses. The series of events that links these two phases together is called excitation-contraction coupling. First, a wave of action potential spread.
Exactly. So you know already how the action potential is created. The action potential is created by the activation of the nicotinic receptor in the sarcolemma. Yes, and this leads to flow of the ions. We are thinking mainly about sodium ions through the cell membrane. It changes the potential of the cellular membrane. Then it would activate the voltage-gated sodium channels, and the voltage-gated sodium channels, as you already know, are responsible for the propagation of the action potential.
It's from the motor end plate in all directions. This is similar to the ripples formed on a pond when a stone is thrown. When this wave of excitation reaches the transverse tubules, which are commonly known as T2.
As you remember, the transverse tubules are those invaginations of the cellular membrane that gets deep into the cell in order to enable fast propagation of the action potential deep inside the muscle cell.
Tubules. It continues down them into the sarcoplasm of the muscle fiber. Next, the action potential stimulates the opening of voltage-gated ion channels in the T tubules. These channels are physically linked to calcium channels in the terminal cisternae of the sarcoplasmic reticulum. These calcium channels open as well. And since calcium ions are higher in concentration in the sarcoplasmic reticulum than they are in the sarcoplasm, they diffuse out and into the cytosol.
Exactly. So this is a very important moment. Yes, we get the calcium ions out into the cytosol, so into the cytoplasm generally.
The calcium ions will now bind to the troponin of the sarcomere's thin filaments.
Okay, so we have a troponin, yes, binded to the tropomyosin, and that tropomyosin is very important substance. Tropomyosin is hiding the myosin binding site. As you probably heard couple times during your life, the most important proteins in the muscle contractions are the myosin and actin. So myosin and actin needs to interact, needs to change relative position. In order to enable this changing of relative position, myosin and actin needs to bind. Yes, obviously. But myosin is binding to the actin in myosin binding sites. And those myosin binding sites are covered by the tropomyosin. So in order to for the myosin to attach to the actin, in order to be able to start contracting, tropomyosin needs to move. And how the movement of the tropomyosin is coordinated? The movement of the tropomyosin is coordinated by troponin. And how troponin is coordinated? Troponin is coordinated by the calcium. Okay, so let's go on. I suppose it would get clearer with the video going on.
The main protein of the
see, first moment, calcium attaches to the troponin. So the troponin would be activated and would do something. Think for a moment, what would be this action?
Filament is called actin. This binding causes the troponin-tropomyosin complex to change shape and move.
Exactly. So it would change shape. And then you see these violet places. Yes? Think for a moment, what is it? What are those violet places on the base of what I already explained? Okay.
Into a groove on the actin. This exposes active sites on the actin filament.
Yeah. So these active sites are the myosin binding sites. Yes, myosin binding sites on the actin. So we are still on the actin, obviously, but these active sites are the sites where the myosin would attach in order to enable muscle contraction.
Active sites are now available for binding to myosin heads, which are the main protein of the thick filament.
Exactly, and as you can see on the picture also, there is ADP and phosphate. Generally, it's because, as you know already from other presentations, ATP is the main substance that conveys energy in the organism. Obviously, muscle contraction uses a lot of energy, so we need a lot of ATP, simple like that. And because of this, on the heads of the myosin, there are it needs constant replenishment of the ATP in order to enable muscle contraction. Okay, I suppose it's quite complicated, but there is one good thing about videos, that you can get couple times through them. So I hope that it will get clear. You can of course ask some questions on the classroom if you want. So, goodbye.